All right. Let's look at this. So last time we saw that we were discussing about transcription and translation. So now we need to look at how does this translation have an application, because in most cases the question come as this. When it comes as this, is just a simple description that describe the process, something like that, without asking you um, many questions concerning about that. Ish.
A is a DNA molecule. And DNA it's mRNA strand, the one in the middle. DNA template or strand. Uh, yes, anticodons. Codes are found in the DNA. Codons in the mRNA, tRNA. They are codons. They are anticodons. It's amino acid. Ah. It's a peptide bond. But they are amino acids, say. It's a peptide bond.
One amino acid plus another amino acid plus another amino acid plus another amino acid. I join them together by a peptide bond. How do you call this structure? Poly? Polypeptide chain. No? Yes, we don't have any polypeptide bond. It's called polypeptide chain. So these are some of the questions you have to understand before you go for anything related to what? Before you go to anything related to uh, uh, before you go to anything related to this topic, you have to understand this before you go, like you go to another topic. So if you don't understand what is a code, what is a codon, what is the anticodon, what are the bonds, or how, then the questions when they come like this, they're gonna kill you. How do you call the bond between, if this is one amino acid proline and valine, but there is a bond in between, how do you call that bond? Say it is it's called what? Peptide bond. Peptide bond. So I think we are done with that. Then we got the next one. You got the next one is saying that. All right. Now provide molecule 1. So now we know now what is molecule 1, isn't it? Let's find out where is molecule 1. What is molecule 1? Molecule 1, we have said that it is what? It's DNA molecule. Then organelle 6. How do you call this organelle 6? Where translation occurs? Ribosome. Give only the number of the part that which represents DNA strand. It's number 2. Number 2. DNA template, because they're saying template. If you look at it here, they're saying what? Template. So because this one is facing this side, it means that messenger is being copied. Is messenger is formed from this strand, not this strand. Okay. What is codon? Something which indicates a codon. 7, because codon, we say that it is found on messenger RNA. Then they're saying describe the process of, what is B? Oh, monomer of protein. They are looking for the monomer. Remember that proteins are made from amino acids. So it means that here we're gonna look at number 5. Yes, number 4. Number 5. Just this. Remember last time we said, I asked a question, how do you call a combination of all? So it's a polypeptide chain, but only one structure in there, like glycine. If you look at the glycine, it is amino acid. When you come here, it is proline, it is amino acid. So it is the monomer. Remember I said that monomers are the building blocks of those structures. They are the building blocks. So basically that is it. So we go to the next question. The next question, what is it saying? Describe translation as it occurs on organelle 6. So this is an open question whereby you just have to describe what happens on organelle 6. So organelle 6, basically you say that if you come back here, they're just looking at the process of translation. According to the messenger RNA codons, the transfer RNA with the complementary codon bring the required amino acid to the ribosome. The amino acids are joined together by a peptide bond to form a required protein. So basically, you just explain what is here. Each transfer RNA carries, you see in the book, each transfer RNA carries a specific amino acid. When the anticodon on the tRNA matches with the codons. You see that anticodons are found on tRNA, while codons are found on the messenger RNA. The moment they match together, now the transfer RNA will bring this amino acid to the what? To this structure which you call the ribosome. Sometimes we just draw a simple circle, sometimes two circles. Yes, so because the ribosome has the major subunit and the minor subunit. You understand? But now, as long as you see that the messenger has a line there, that is a ribosome. So the moment the anticodon matches with the codon, now the transfer RNA will bring the required amino acid to this ribosome. Now the amino acids, they are being attached together by peptide bond. I said, please don't forget this, that peptide bonds are peptide bonds. Sandion, peptide bonds, we don't say polypeptide bond. We don't have that. When you add the word polypeptide, peptide, it means that you are trying to describe the chain, the polypeptide chain. The moment you start with the word poly, you are trying to describe the chain. So the bond, we call it peptide bond if it's one. If there are many, we call them peptide bonds. Yeah, so the moment you add the word poly, you're gonna cross it. So these amino acids are joined together by these bonds, which you call the peptide bonds, to form a required protein. So I think that one is done. You have to know that. You want it or not, you have to know that before you go for the paper. So we got the next question. There's provide a DNA sequence code for glycine. Where is it? DNA sequence code for glycine. We come back here and then we look for glycine. So if you look at glycine, this is transfer RNA and codon. This is messenger RNA codon. So they are looking for DNA of which I'm gonna say G goes with the C goes with the still G goes with the C, and then A will go with T. Why? Because A is in messenger RNA or in RNA, A goes with U, while while in DNA, A goes with T. Is it clear? Yes. Then we got the next question. They are saying that describe the sequence that okay, we're done that. The codon for proline. When they say codon for proline, what does it mean?
They want the codon from mRNA.
Yeah, we are looking for the codon for
mRNA.
Yeah, remember that codons are found on RNA, isn't it? Yeah, the moment we are finding them on RNA, then automatically we have to talk about it. We have to talk about it as what? As the messenger RNA. Therefore, we come back here, they're saying for proline. Proline is here, so we are looking for this. So the answer is going to be CCU. We go. State two difference between DNA nucleotide and DNA and RNA nucleotide. We have discussed about these questions. Sandion, give me two difference between DNA nucleotide and the RNA nucleotide. One for Sandion, one for Zintle. Zintle, start.
Okay, DNA has a sugar called deoxyribose, and number two, DNA.
I want only one. I only want one. Wait. I only want one.
Oh.
Yes. You said DNA has
a sugar called deoxyribose.
DNA has a sugar called deoxyribose. Okay. RNA has what?
You asking me, sir?
Yes. Okay, now if you say DNA has deoxyribose, what does RNA have?
RNA has a sugar called
ribose. Sandion, what's the next point?
DNA has nitrogenous base trimethylene.
saying that DNA has nitrogenous base thymine, while RNA has nitrogenous base uracil. Yeah, so those are the two differences you can talk about when it comes to the nucleotide. They are not asking the whole structure of DNA, they're only focusing on the structure of the nucleotide. And since they said just state the difference, even if you write it in paragraph, it's fine. Unless they say tabulate. When you say tabulate, that's when you have to draw the table. And when you're drawing a table, it must be a complete table, not just a fake table. It must be a complete table, a complete table. Then you write here and you write here. Then, yeah, you write the caption that this is DNA and this is RNA nucleotide. Then after that, you write the points. And this point, if you talk about uracil this side as a base, you talk about thymine on the other side as a base. You don't mix them up. Right. So we go to the next question. So we are entering into that zone which I wanted. They are saying that the sequence of amino acid in a protein molecule is coded by DNA and messenger and RNA. The table below shows messenger RNA code and the corresponding amino acids. All right. According to the table, how many codons code for phenylalanine? If you look at this table, let me zoom it out. They're saying that how many codons code for phenylalanine. So we need to find out which one is phenylalanine. They're saying codons. This is messenger, then automatically. So you have this phenylalanine, that's one, and then you have this phenylalanine, that is two. Is there any other phenylalanine? No. So it means that you have one, and I also have two. Is there any other phenylalanine? No. So it means that our answer is going to be 2. What is the anticodon for glutamic acid? Glutamic acid is here. But they are saying what is the anticodon? Because this is messenger RNA. Are you seeing? Because this one is messenger RNA. So if this is messenger RNA and they are looking for anticodon here, so what does it mean? It means that I have to convert this, because this is messenger RNA, into transfer RNA. So G goes with the C, A goes with the U, G goes with the C. So the answer is going to be CUC. I hope you guys you got those answers. If you got a different answer which is not what we are getting now, you cross it and then you do the correction. They're saying that a section of messenger RNA has the following base sequence and it is read from left to right. Okay, we go to left to right, from left to right. Alright, what are they saying? DNA triplet for the last codon on this messenger RNA. Yes, the last triplet. They are saying triplet for the last codon. Because this is messenger RNA, yes, this codon, this structure is a messenger RNA and also indicated by U. They want a triplet for DNA. You see, DNA based triplet, meaning that I have to come here and I convert this into DNA. So A goes with the T, C goes with the G, and another C goes with the G. So the answer is gonna be T, G, G. Is it clear? We got the next one. The first amino acid coded for by this section of messenger RNA. The first amino acid, meaning that we are looking for this. So how do we call that? Because this is messenger, and then we need to come back here and find out what is here in the table. What is in the table? What is in the table is a messenger. You see, it's a messenger, and what is here? Where is it? DNA triplet for the last codon. What is on the message? First amino acid coded by this messenger. So they are looking for the first amino acid. Yes, look here. This is a messenger. When they say a section of messenger, also this is a messenger. They want messenger. They gave us messenger here. Also here they gave us messenger. Do I need to convert? No. So I'm going to have to read it the way it is. GUA. So I've come here and have to look for GU. Is it GUA? GAU. GAU. GAU, GAU, GAU is here. Which is it? Aspartate. Yes. It's aspartate. So the answer is supposed to be aspartate. Zintle, did you get it right?
Yes, sir.
But Zintle, when you are copying me, ne?
No, I was not copying, sir.
Uh-uh. Because you are getting everything right. I'm not saying that you must get it wrong, but I'm just saying that something is fishy now. All right. Uh-huh. Then we got the next question. Should we continue?
Yes, sir.
A mutation occurred when the result in the following base sequence on messenger RNA molecule. Isn't it? Then you're saying that. Oh, we are done with that. A mutation so they're saying that a mutation occurred. And now we need to find out where did the mutation occur. You know, I told you that when it comes to mutations, yeah, stories will start. Okay, I have this, 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 I have. Oh, look at that and look at that. I have this, I have this, I have this, I have this. So it means that 1, 2, 3, 4. It means that the mutation occurred on the on codon number 4 on the third base, because here it shows that the base was C while it is replaced by U, isn't it? So describe the mutation that occurred here. So when you look at that, we shall see that I need to find out, does the mutation code for a different amino acid or it codes for the same amino acid? How do I find out that? Because this is messenger and this is messenger, I don't need to change anything. I will have to read it the way it is. So AGC is what? Let me first clear up here. AGC, did I say it right? Yes, AGC is serine, isn't it? But it changed to AGU. AGU is what? So I have to look for, so we say this is serine, AGU. Is it AGU? Not GAU, ne? AGU. AGU is also serine. You understand? It means that the mutation has changed, but it codes the mutation. The base has changed. The bases have changed in the sequence, but they code for the same amino acid. Will the protein have any effect? Will there be any effect on the protein? No, because it codes for the same amino acid. As I said that you can be Zintle Karabo. However much those names they are not together because different cultures, but it's fine. Just let's use it as an example. You are Zintle Karabo, meaning that when Zintle, I can call you Zintle, you say yes sir. I can call you Karabo, you say yes sir. Why? I just changed the name, but it remains the same person. Does it mean that you're gonna change the structure? No. So it is the same thing. It's the same thing. However much the bases have changed, but they code for the same amino acid. Therefore, the protein will not change. So, since they are saying that, describe the mutation that occurred. First of all, when you say the mutation which occurred, now we are trying to see the answers. The mutation which occurred is the base on the 1, 2, 3, 4. codon number 4. Yes, the third base changed from C to U. That is the mutation. They're asking you what really happened. That's what they're asking. So the code, the base codon number 4, yes, 1 2 3 4, yes, the third base of that codon number 4 changed from C to U. So yes, then they're saying that explain the effect, explain the effect, explain the effect that the mutation described in the question above will have on the resultant protein or the resulting protein. So we have seen that the protein will not change because it coded for the same amino acid. You understand? So the resulting protein will not have any effect. Yes, or this mutation will not have any effect on the protein. Why? Because we said because the two codons, they code for the same amino acid. Then you mention that amino acid. Which amino acid is that? And that is serine. Zintle, are we together?
Yes, sir.
Yeah, this question, when they bring it, in most cases, people fail it. People fail it, and there's nothing we can do. And we know that if you fail something, we're gonna keep bringing it until we see that you guys, you have got it right. So guys, make sure that you should not miss this out.
Yeah.
We got the next question. um um Yes, today's class we made it live, so people are on YouTube and on TikTok. Yeah, you must tell us what's your problem as we continue. However, if you want the classes to continue like that, you have to register with us. All right, let's continue to the next question. Name and describe the process occurring in the nucleus which result in the formation of messenger RNA. Name and describe the process. They said name. You mentioning the name. When you give the name, automatically you get 1 mark. The name of that. Rorisang, you are not talking anything. Are you there? Okay. You're saying that. Name and describe the process that occur during in the nucleus. You see, this is the location. So what are they looking for, which result in the formation of messenger?
Oh.
What are they looking for? Transcription. So you mentioning that this is process called transcription, yes, it's a mark. That's 1 mark. So now you have 5 marks to go. Now from there, you start to describe the process of transcription. So you have to first identify it correctly, because if you describe translation or if you say translation, then you're gonna get it wrong, because translation occurs in the cytoplasm on the ribosome. The process which occurs in the nucleus, it is transcription. And number 2, another process is DNA replication. Those are the only 2 processes we talk about on our curriculum. Those are the 2 processes. So when they talk about the nucleus, what must come into your mind? It must be DNA replication and transcription. But since we are on transcription, on protein synthesis, so definitely the answer is transcription. And they even specified that which leads to the formation of the messenger. And I say that the process by which messenger is formed is called transcription. Yes. That's why even here it is indicated here that they say that transcription is the formation of messenger RNA. Yes. So I think we are done with that. So we go to the next question. It's the last question on this so that we go to meiosis, isn't it? Let's just use very few minutes because I think we have ever done this. Saying that what is W? Let's mention it very fast. Let's mention it. Let's mention it very fast. What's W? DNA. DNA molecule. What is the X?
X is
X is messenger RNA. What is the Y?
amino acid.
Y is amino acid. What is Z?
Ribosome.
Ribosome. What is this?
Polypeptide chain.
Poly? Polypeptide chain. And then what is this?
Transfer RNA.
Transfer RNA. And what's the bond between different amino acids?
Peptide bond.
Peptide.
Peptide bond. Not
Not polypeptide.
not polypeptide bond. Please do not you say polypeptide bond. It's peptide bond. Thank you. How are the bases on the transfer RNA? 3 bases, how do you call them?
Yeah.
These 3 bases, these bases on transfer RNA, how do you call them? Anticodons. 3 bases on messenger? Codon. Okay. All right, let's go. Identify the molecule W. DNA. D? DNA molecule. What is Y? We said what molecule Y?
Molecule.
Y. Yes.
You said amino acid.
Amino acid. We continue. Okay. Then they're saying that name the part of the protein synthesis indicated by A. So they're looking for this.
It's transcription.
Transcription. Isn't it? Yes. Kidwa. Kidwa. What's the problem, Kidwa? All right. Name the part of protein synthesis indicated. Okay, we're done with that. Describe how mutation on W will affect the structure on B. Describe the that is another mutation, you see. If they're asking you to describe how a mutation on W, how is this mutation going to affect the structure? This structure, the structure formed by process, meaning that this. So if you have a change in the DNA sequence, will result in a different sequence of messenger, isn't it? Because DNA, look, DNA forms messenger, isn't it? DNA forms messenger. If DNA has changed, what will happen to the messenger? It will change. If messenger has changed, what will happen to the sequence of these amino acids? They will also change. If the sequence of the amino acid have changed, what will happen to the protein? The protein will also change. So if the DNA is changed, the sequence of messenger will also change. It means that different amino acids will be brought to the ribosome, yes, in a different sequence, isn't it? Resulting in a different protein. Have you got it?
No.
So you are writing. No, you just need to understand because we always bring these questions in a twisted way. We can start from messenger and ask you what will happen to this. We can start from DNA and ask you what will happen to this. We can only talk about DNA. We ask you what will happen on this. So you just have to know basically what is it. Is it clear? So a change in the DNA sequence, yes, will result a change in the DNA sequence will result in a different sequence of messenger RNA, isn't it? Which will result in different amino acids brought to the ribosome. hence a different protein or different polypeptide chain and a different protein. So is when DNA changes, messenger RNA will change. When the messenger RNA changes, the amino acid sequence will change. If the amino acid sequence changes, then automatically a different protein will be formed. So that's the idea.
So, sir, still, why should we? There's no different protein.
There is no what?
It's all the same.
No, the proteins are there. They are different. They are there. We have different protein.
So the protein are different because of mutation.
Because of what? If you change from that protein to another protein, because a mutation means change in the sequence of what? Of that the sequence of DNA, and DNA is the one which codes for the protein which is being formed. So if you change the sequence, then automatically a different protein will form. An example is the protein for skin color. Yes, if you have a different protein for skin color, what will happen?
Skin color.
Yes, what is going to happen?
You won't have the same color.
You won't have the same color. You won't have the same color. So guys who are online, you're gonna just have a short break so that we come back for, yeah, people who are online, you can do that question. In 14.4, they're saying that describe describe how mutation. We are done with that. They're saying that the following sequence the following sequence represent a part of nitrogenous base sequence, a nitrogenous base sequence on molecule X. Okay, if they're saying that you have triplet 1, triplet 2, triplet 3, isn't it?
Recording in progress.
Write down the base sequence of anticodon. This, you see now, anticodon of triplet 1. If they say anticodon, what are they looking for? tRNA.
Yes.
They're saying that the following sequence represents, okay, base on molecule X. Which way is molecule X? Molecule X is messenger RNA, meaning that what is here is messenger RNA. So if they're asking us transfer RNA, then you automatically have to convert it. All right. So what is the next? The table below shows the amino acid that correspond with the DNA. You see, they are saying now corresponding with the DNA. So you have messenger, you have tRNA, you have DNA. What do they want? Okay, let's find out the question. What are they talking about in the question?
Write down the correct. Yes.
Write down the correct sequence of amino acids. They are saying amino acid for triplet number 1 to number 3. For triplet number 1 to number 3. This is the different question. Write down the base sequence of anticodon of triplet 1. So it means that because this is messenger and this is transfer RNA, therefore I just need to convert this. Is A goes with U, G goes with C, A goes with U. So that is the first answer. Then they are saying that write down the correct sequence of amino acid for triplet number 1 to 3, but they gave you DNA, meaning that what I'm going to do is, because this is messenger and then this is DNA, I have to convert the messenger into DNA, because what is here is DNA and what is given is messenger. Therefore, I have to convert this messenger and then now I put it into DNA. So what am I going to do? I'm gonna say it's gonna be U C U, isn't it? Then I look for U. Sorry, for DNA, not you're converting into DNA. So it's gonna be T C T, because you are converting it from messenger RNA to DNA. So TCT is what? So is arginine. So the first amino acid I'm gonna write is arginine. Then the second amino acid is T. U goes with the A, and then G goes with the C. So it's gonna be TAC, which is methionine. Yes, and then automatically the last one, just to verify, is gonna be CCT. So which is the CCT, which is the glycine. So basically, this is what you're supposed to write, but you write it in full so that you're able to get the answer. So you get it right.
One, I will, one,
You wrote what?
title 1, answer 9, I'm sure.
So you wrote them. So if you wrote them, as long as you got them right, then at least you have the mark. Then we go to the next question. So that is it, isn't it? That is it. It means that we are going to the meiosis, and then we see what we can do with what? With meiosis. Yes, we're gonna just do a simple highlight, and then after that we can link it to what? To the questions which are most likely to be asked, or sample questions. Okay. So meiosis. So when it comes to meiosis, there are some things which you need to understand. Number one, what is meiosis for? What is it used for? Remember, meiosis is a type of cell division where one cell divides to form two cells. After that, the two cells divide to form four cells. And then this one comes as 23 pairs, meaning that 23 pairs of chromosome, meaning that it's 2n, which is equivalent to 46 chromosome. Then when meiosis 1 occurs, it's going to be 23 chromosomes. They're no longer pairs, and then when this one split, then it's going to become single-stranded chromosomes, which is 23 here, 23 here, 23 here, 23 here. But what you need to know, meiosis 2, what you need to know that all of these are different from one another. They are different from one another. So it means that if this is a sperm, this is a sperm, this is a sperm, this is a sperm, all of them must be different sperms. That's why if different sperms they fertilize different ovums, though you end up producing non-identical twins. It's because of the different sperms which are fertilizing the ovum. So now what's the function of meiosis? Basically, the major function of meiosis is to produce haploid gametes. Number two is to produce what we call variation. Yeah, and not just variation, it's better to say genetic variation. Yes. Then another answer you can talk about is to maintain the chromosome number from generation to generation. Why like that? Is because during fertilization, the father brings 23 chromosomes, the mother brings 23 chromosomes. When they combine, you form 46. So meiosis is supposed to reduce them back to 23 so that the number of chromosomes in the individual is kept constant. So that is the meaning of that. So when you're done with that, this is what they are trying to say. Where do we find meiosis? So where do we find meiosis? We'll find out that meiosis, where do we find meiosis? We shall say that meiosis in male, you'll find it in the testis or testis, either way, because this is singular, this is plural. And then you also find it in the pollen, the pollen grains. Yeah, these are animals, these are plants. And then meiosis in female, you find it in the ovaries in animals and ovaries in plants. So if you look at the plants, the animals, they will produce sperms, while plants, they will produce pollen grains. Did I say it's found in pollen grains? No, it's found in anthers. Yes, sorry for that. It is found in anthers to produce pollen grains. So the sperms of the plants, they are pollen grains. So it results in the formation of pollen grains. While in animals, it is ovum. If there are many, we call them ova. And then in plants, it's ovules. So this is what we are trying to say here, that in animals you'll find it in the ovary to produce ova. In plants, so in male, you'll find it in testes to produce spermatozoa, which are the sperm cells. And then in ovary, in plants, to produce ovules, and then anthers to produce pollen grains. So that's what you need to know, where is it located. And then we said the functions of meiosis. Yes, we will see it when we look at this. Yes, we see that the function of meiosis at the end here, the importance of meiosis, that is to produce haploid sperm cells, that is haploid gametes. Those are the sperms and ova. And then number two, the halving effect overcomes the doubling effect. That's what I'm saying, that it maintains the chromosome number constant from generation to generation. So it has that. Then another one is to cause genetic variation. That is very, 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 very important. You have to know that it causes what? Genetic variation. Thing has refused. Okay, then another one is haploid gametes, genetic variation, and then also maintaining the chromosome number from generation to generation. Those are the major functions of what? Of meiosis. So what makes meiosis to be complicated? It's a simple thing. It's a simple what? Simple thing. This generation, these phases. So it's simple. When you say metaphase, What you need to know is IPMAT, isn't it? IPMAT. Ne? Interphase, prophase, metaphase, anaphase, telophase. Interphase is what you call, I always call it growth phase. That's when it's growing. And then this is the reproductive cycle. So prophase is that we call the preparation. They are preparing themselves. Metaphase means they're in the middle. So if the chromosomes are like this, look, you must always look at it like this. When it's straight, ne? Because this is where the problem starts from. You see this, you see these are the centrioles, ne? And then these are the chromosomes. Yes, they're in the equator, ne? So this thing must always be inside. Sometimes when we bring it, we bring the like this.
Okay.
And then now when you get lost, where am I? Where is the equator? It's because now this thing is up, now this one up, this is down. Just turn your paper, ne? So that they face, these things must be on the sides. The centrioles must always be in the sides. So that's when you see that, oh, the chromosomes are in the middle. So, and then when you say anaphase, there must be a space in between. If it is chromatids, must have a space. If it is chromosomes, if they are chromosomes, you see, you can't say that this is what? Is metaphase. Why? Because you have a big space in here. So that space is what you call the anaphase, which means apart. That's what you call the anaphase, which means what? Apart. So once you know that, then at least you have identified the phases. Then telophase, automatically they have reached the terminal. But there are some few things which you need also to take note of this, that when it comes to meiosis 1, these chromosomes are always in pair. They are full chromosomes. While when you look at the same phase of anaphase, you'll find out that it's just these chromatids which are being pulled. You see, it's just, you see, half of a chromosome is being what? Not half, one side of a chromosome. You see, it's not full chromosome like this. So there's also space in between, but if you look at the chromosomes here, the chromosomes, they are full one. They have two strands. While here, it's only one strand being pulled. The same story here, the same story here. So you have to know, because this is where you start losing the answers there. So then another thing where you have issues, it's when we come to the karyotype. When we ask you questions concerning about karyotypes, and then you end up losing it, you don't know which one is a male, which one is a female, how many chromosomes. We're gonna reach there and then also see. So let's do some few questions about meiosis, and then we see we can also link it. Okay, they're saying that the diagram below shows the arrangement of two pairs of homologous chromosomes. Yes, two pairs of homologous chromosomes in the cell. This is homologous to this. This is homologous to this. But now the question is asked, which phase is this? Anaphase, because there is a space in between. Centrioles are that side. So it means that is anaphase, but because they are homologous chromosomes, therefore it's going to be anaphase 1, because we only find this in meiosis 1. So it's anaphase 1. Name 2 characteristics of homologous chromosomes. What can you say?
Same shape. Can I say shape?
They have the what?
Same structure. Same structure.
Same structure?
The same height.
Same height. Same size. They have the same size, the same shape, and the same genetic composition. Or we can say that same.
Okay, shape and size.
No, same size, same shape. That one is obvious. Then there is another one which we say that they have the same location of genes. Yes, meaning that if you have the location of gene for height or for skin color, in this chromosome, it must be in the same location as the other chromosome. So, yeah, that's why I'm saying that same locus of genes, yes. So, so we have said that they have the same size. You understand? Same location of genes. That's why you see that. Now, the white one,
Height is not correct.
the white one is the same thing here. You understand? They are not the same. They are not the same. If they are the same, then we wouldn't have been different. And if crossing over takes place, then we would have been no genetic variation if they were exactly the same. The what is inside them could be different, but the location of these genes is the same. So that's why I'm saying that same location of genes. So identify structure A. What is structure A?
What is A?
It's this
Spindle fiber.
spindle fibers. Spindle fibers. And they are saying that how many each of the following is present in the diagram. That's where the problem starts from.
Of each. Oh. Okay.
Yeah, that's where the problem starts from. Chromatids.
There's 4.
How many chromatids? How many each of the following? There are 4 chromatids.
How many?
Remember, the chromosome is formed by what? By 2 chromatids.
2.
So how many chromatids are shown? You have 1 chromatid, 2 chromatids, 3 chromatids, 4 chromatids, 5 chromatids, 6, 7, 8. So there are 8 chromatids because each chromosome it is made up of 1 chromatid. Not 1 chromatid, 2 chromatids. Then they're saying that how many each of the following is present. The moment they start asking how many each of the following, now students will go off. Centromere. How many centromeres do we have? Because each chromosome is made up of 1 centromere. You understand? Yes. So each chromosome, 1 centromere. Each chromosome. So draw the diagram to show one of the cell that would be formed at the end of telophase 1.
One of the cell.
Yes, they're saying at the end of telophase 1. So I expect you to, this becomes a cell, and also this becomes a what? A cell.
So they are both a cell.
They are going to be both cells, but they are saying telophase 1 at the end of telophase 1. So it means that what am I going to do is so you're going to have
Recording stopped.
You're gonna have 4.
you're going to have 4. You're going to have 4. Just a moment. Yes, you're going to have how many?
4.
You're going to have 4. You're going to have 4. No, at the end of telophase 1. You can't have 4.
Oh, 2.
Just a moment. Ish. Okay. Okay. It's fine. I'll sort it out. So it's gonna be how many? They're going to be 2. 2. They're going to be 2 cells. But now check the variations. Check the variations. So this is gonna be on this. So it means that this cell is gonna have their cytoplasm is gonna have its nucleus. You understand? but these variations are supposed to be indicated. Let me draw it here.
As family was 6.
Yes, you're gonna label. So you draw a cell, you draw the 2, I see, and then you shade the way it is shaded there. Then you have now this one.
So I can take any of the.
Actually, this and this, they are going to one side. So, but now if I draw it like this, I might get it wrong. Why? Because the second one is shorter than the first one, you see. But now the variation is down. So it means that I'm going to have that and also that. Then the remaining one I shade, and then here I shade. You see how it looks like? Then now if this is, now I put the cell membrane, I put the nuclear membrane, then I put the chromosome. They are not homologous because they are different sizes. I put the chromatid. Okay, one is a chromatid. The whole thing is a chromosome. Then you have the centromere. You see? So, and after that you put the caption. So here the 6 marks will come from the caption, correct? Will come from the variations, you shaded them correctly. Another mark, it will go to the number of chromosomes you have drawn. It would be the number of chromosomes you have drawn, and you have drawn the correct phase, because you might draw a wrong phase. Yes, it's a mark. And then now they might also give some 3 from what, from labeling, or some 2 parts which are labeled correctly, so you get the mark. Let's just do one question, and then yeah, I give you homework. Want to do this one? Still have a lot of questions. You can't finish them. Okay, let's do it. They're saying that the diagram below represents the meiosis. Cell 1 undergoes division to give rise to 2 and 3. Some alleles are indicated by the letters. Okay. Let's start. Point number 1. Explain why cell 1 does not belong to a human cell. Why is this thing not belonging to a human cell?
Cell 1 has. Why it doesn't belong to me?
Why doesn't? You first ask yourself. You ask yourself, you go back. How many chromosomes does a human cell have?
6.
46. Okay. How many chromosomes do you have here?
4.
1, 2, 3, 4.
So it doesn't.
It does not belong to human cell because it has 4 chromosomes.
The one that we did before, it doesn't belong also to humans.
Yeah, they didn't say that this is a human cell. They only, unless they link it to a human cell. These cells we do, they are not from human. In most cases, the cells and the genetics we are using, they belong to fruit flies, which you call the Drosophila melanogaster. Yes. So the reason why we use them, because they have a thick chromosome. Their chromosomes are big, so they're able to be visible. So that's why we use them. So it's not a human cell because it has 4 chromosomes instead of 46. How many chromosomes would there be present? You see now, they are giving assumptions. Would be present if cell 2 cell 2, this one, ended cell 2 at the end of telophase 1. So it means that it's gonna divide. This one becomes a cell, this one becomes a cell, isn't it? So these things are going to go to that cell. These things, chromosomes, yes, are going to go there. So how many will there be at the end of telophase 1? So the total cell, the number of chromosomes, there are 4. But here we say that at the end of meiosis 1, you have half, because you started with 23 pairs of 46 chromosomes, but now you have 23. So it means that it's going to be half of it, isn't it? How many chromosomes there are present at the end, cell 2? So it's going to be 2. What are cells produced by cell 3 after meiosis 2? It's going to be 4. At the end of,
So those are
wait, you are saying that daughter cell produced by cell 3 after meiosis 2. They are talking about only this.
cell phase.
So this cell 3 is gonna produce how many? We go back.
It's gonna produce.
You have cell A. Wait. You have cell 2 and cell 3, isn't it? We come back here. This is cell 2. This is cell 3. This is cell 1, isn't it? Based on this, you have cell 1, which is on this one. Cell 1. This cell 1, isn't it? Cell 1 here. Then produces cell 2 and cell 3. So they're asking you how many. So this is cell 1, this is cell 2, this is cell 3. How many will there be present at the end of this? We said there are 2. So they produce 2 and 3. So now they're asking you how many cells will 3 produce at the end of telophase 2?
1.
This 3, this cell 3 will produce how many cells? 2. So that's what they're asking you, that this, how many chromosomes will there be produced? Only cell 3, not the whole cell. So the answer is what? Is 2, not 4. So
At the end of meiosis 2.
yes, but they're saying produced by cell 3. Cell 3.
I always think now the end of meiosis 2 is telophase. So
Yes, they're always 4 if they're talking about cell 1. Okay, if they are 4, if this one produces 4, what about this?
0.
You see? So this one produces 2, also this produces 2. So when I add them, they form 4. Draw a diagram of a gamete that will result in meiosis in cell 2. So now here also the variations are important. The variations are important.
Because the same thing that we know.
Yes, but now you have to show this.
What does it mean
This.
for R?
No, it's just a gene. They just gave you so that they see that you do it right. Because now if you write capital T here, small t here, small t, yet small t has gone to that side, you're gonna get it wrong. It's just to show the variations. So you're gonna draw this for me and then you send it to me so that I'm able to check and see if you have done it right. So I think today we're gonna stop here. I'm gonna send you some activities. Make sure that you send me those activities maybe before you come back for the class, so that you are able to see.
Before Saturday.
Hey, we have no. When you say before you come back to class, I mean before we have another class, yeah, which is Monday. Yes. So make sure that you do it. So guys who are online, thank you so much for joining us. If you want more videos, you can check them there on our channel. See you again in the next class. You have any question?